Good morning, group. Good morning, Dr. Faison. Hope everybody having a great day. This is Malik Dockery speaking on cell adhesion. Let me share my screen with you all first and foremost so that you all can be able to see my PowerPoint presentation. And let's get started. So I'm here to talk about cell adhesion. So what is cell adhesion? Cell adhesion is the ability for individual cells to associate with one another from tissue, organs, and organ systems. Now we have different type of cell adhesion molecules. We have caterins, we have selectins, and we also have integrins. And while we'll be discussing all of those different type of molecules as we continue through the PowerPoint presentation. So we do have different type of tissues in cell adhesion. We have connective tissues and we have the epithelial tissue. So as for the epithelial tissue, it's the outer layer of the skin, and um, cell adhesion is going to hold all these cells in the correct layer. And before I continue, this is the junctions of this extracellular matrix. We do have the apical region and we do have the basolateral region as well. So let me discuss the basal lamina a little bit. The basal lamina holds the tissue in place. So this holds all of this in the correct order. And then also they keep the basal lamina keeps the cells in their correct junctions, I would say. Um, so as for this figure right here, you see that we have the extracellular matrix. Um, as for the extracellular matrix, it makes protein and sends them out of the cell. So you see the different type of proteins around the cell, and the connective tissue is loose. So these purple little cells around the extracellular matrix is protein, and once these proteins are then created, they were going to enter this. They will be sent out of the cell. Let's continue. So we have different type of cell junctions. We have adhesives junction, tight junctions, and gap junctions. So as for the adhesive junction, it's just a cell that is connected to the cytoskeleton by the microfilaments, and we then have the tight junctions that keeps the animal cells tight and prevent molecules from entering the epithelial cell larger through epithelial cells layer through the spaces between the adjoining cells. Also in the gap junction, it's just a cell that's connected between two adjacent cells so that ions and small molecules will be able to pass. So, as for this figure, you see that here are the tight junctions. You can tell that they are really bonded really tight to make sure that nothing enters this area down here in the basolateral region. These are the gap junctions in this figure. As you can see, those are one of the cell junctions in animal cells. And you see the desmosums, and which is these purple figures is hit right here. Also, you do see again the basal lamina and the connective tissues that is also in this figure as well. This is very important in the apical region. This makes sure that any fluid or any and basically really enter fluid, make sure it don't enter the basolateral region, and make sure everything stays function within the skin. So let's continue. So now we're here at this table. Um, in this table, you see that it has the main function of each of the different type of adhesive junctions. And you see it talks about the decimal sums, tight junctions, the gap junctions, etc. It also tells you about the spaces that it covers and the associated structures as well that is connected to these different type of junctions, so that you can get and understand more knowledge of um, cells that are in cell adhesion. So now we have our cadherin structure. Inside of our cadherin structure, the cadherins play a major role in cell recognition and cell adhesion. Also, cadherins require Ca2 plus in order to bind and regulate cell adhesion. Um, so here you see that you have the E catenins, you have the beta catenin, you have the alpha catenin, you have the actin inside of the cell, and then you have the E catenin on the outside of the cell. Um, so this um, basically the green, all the highlighted green areas are the E catenins, and in this figure, you see the E catenins is holding all the cells in this figure together and around the plasma membrane. So, like I said, these blue highlighted figures are the cells, and everything that's not in blue is the E catenins or is connected to the plasma membrane. So, let's continue. So we have the caterans on cell adhesion. We have different type of caterans. As you can see, we have the E caterans, the epithelial tissue. We have the P caterans, the placental tissues. We have the N caterans, the neural tissue, which are all the different types of cataracts involved in cell adhesion. Now, in this figure, you can see that the effect of cataracts on cells. In the red, you will be able to see the P cataracts. In the green section, you will see that you will have the E cataracts expressing. And one thing is something that you do notice is that these cells rather bind to their own cells. So what I mean by that is you notice that the red P cataracts are binding to the also other red P cataracts in this figure, and in this figure you also see that the E cataracts are binding to the green E cataracts, and they will prefer to bind to their own I would say type, as you would say. They don't really like to um, have E caterans bound with P caterans or interchange with one another. So this is where the homophilic and the heterophilic interaction come about. The homophilic interaction is with the same cells that bind to one another, and the heterophilic interactions with the different type of cells as well. So they wouldn't mind binding to different type of groups or families as well. So we do have the catarins during the embryonic development. So in this figure, you see the breakdown of an embryonic development, whereas the dynamic assembly and this assembly catarins are tightly regulated. So what I mean by that is you see that in this figure that the that you see that these categories are tightly regulated, and these categories on this side are deficient or basically are loose. Um, so sometimes, as you know, well, as you can see, that the dynamic assembly and the assembly of categories is basically really tightly regulated. Other categories help neuron cells form bundles and establish synaptic connections. Um, so as we continue moving forward in the epithelial mesench- mesenchymal transition, this is basically accommodated by um, the changes in calories expressions, and changes in calorie expressions occur in cancer. Like I said, because when cancer cells start producing e-calories, the cells undergo epithelial mesenchymal transition, and the healthy cells will not. So these cells will spread to other parts of the body, known as metastasis, and I mean, excuse me, metastasis, and this will have an effect on humans in their bodies as well as when it comes to cancer. So we also have the desmosomes structures, and it's one type of adhesion between two epithelial cells. It's a type of intercellular junction that holds cells together. So um, in this figure, you see the desmosomes structure is holding the joining cells together, but the caterins in the desmosomes will will excuse me, the caterins in desmosomes are called decimal colons and decimal glycans. In the cytosol, the desmosomes caterins interact with the protein as well. So this is the desmosomes right here. These are connected, and that holds together the cells. As well, you can see it here, this thick layer right here, and also you can see the decimal somial caterins in this figure as well. As we continue, so we have the different extracellular matrix. The extracellular matrix is basically fills in the spaces between cells and have a mixture of proteins. So in this figure, you can see extracellular matrix play a role in shaping the mechanical properties of organs and tissues. These extracellular matrix molecules consist of structural proteins, uh, collagens, and elastogens. Gives help to the extracellular matrix. Second, you will see the protein polysaccharide that provide embedded structural molecules and adhesive glycoproteins such as fibronectins and laminins, which allow cells to connect to the matrix. So these are the cells that are connected to the matrix. Here are the cartilage cells, and that is also the embedded structure of molecule. So this molecule, like I said, is embedded with the cartilage cell, and then here we have basically the role and shape in showing the organs and tissues in the extracellular matrix. So Just a little bit about integrins. Integrins are the receptor that binds to the extracellular matrix proteins. Also, it has two large polypeptides, the alpha and the beta subunit. And as we continue, so here we have this figure, and within this figure is the the fibronectin receptor. So the fibronectin receptor is found in the extracellular matrix and is connected to the cell surface. It's important. It's important in determining cell shapes and cell migration. So in this figure, you see that we have the alpha. You see we have the alpha, we have the beta, we have the outer membrane surface, the plasma membrane, and the cytoplasmic membrane surface as well. So, like I said, so as you see the fibronectin receptor, that it has a binding site for fibronectin on the outside of the surface and binds sites for tallien on the cytoplasmic um, side of the membrane. So the integrins alpha and beta are split into two different segments. This the this fluid this fluid bond is holding the two integrins together on the outer membrane surface. It has a binding site for the cytoskeletal protein. The tails of which is the tails. What I mean by the tails is the tails down here. The tails of the integrins interact with proteins in the cytosol, with which link integrins to the cytoskeleton protein. And now we're gonna go here to the focal adhesion, and focal adhesion is attached to the extracellular migratory and non-epithelial cells. Cells, cancer cells has activated focal adhesion kinase when they are not attached and they are created into cancer cells. Um, and now we're going to discuss this figure over here, and this basically shows in this figure you can see a bunch of different type of migrating cells attached to the extracellular matrix. You see focal adhesion containing integrins that are like proteins in such as penicillin and alpha actinin. So as you see in this figure here, you see that we have the alpha actin, we have the actin filament, and we have the Tylen and the penicillin. You can see that they are showing the different type of integrins, and you can see which part each cell membrane or extracellular matrix or the cytosol is a part of. You see that we have different type of figures. We have the focal adhesion, we have the immunofluorescence, we have the hemodynamism, and we have the electron myograph. Um, and Also, you will find focal adhesion kinase grow promoting, and in the integrin linked kinase, you see scaffolding proteins. And also, you can tell that we have the intermediate filaments in the bottom of this figure. This is where the plaquin family protein, the BPGA1, and we have our different matrices, and we also still again have the integrins. If we can go back to this figure, we can see that the alpha and the beta, like I said previously, definitely plays a major role in the fibronectin receptor, and these is definitely in between the plasma membrane to keep this um, determining like which cells will go through, and also let us see the different type of um, bindings that they have, such as like the binding site for RGD sequence for the fibronectin. Then we, like I said in the video before, I mean, in previously, the binding sites for the Italian and the tails. So this actually concludes my video. Thank you all for watching. I hope I was a great help to you all, and thank you and have a great day.